test tubes for our tank. What's number two? Number two is the thing we're actually going to be doing the separation on. What do we call that? Notice the spots are moving up it. What do we call that part, number two?
Chromatography strips.
A chromatography strip, or another word we use for it is stationary phase. Where do you think the term stationary comes from? And it's not just because it's paper, but stationary phase means that it's not moving, right? So the paper or whatever it is you're doing the chromatography on, it can be a silica plate, it can be a lot of different things. We're going to be using filter paper today. Basically means that it is just standing still and the other things are moving. It is not moving. What is number three? Number three is basically a line on the paper. What do we call that? That's something that you're going to need to be marking today in pencil. What is that? When you finish your run, you're going to mark that with a pencil to see how far something has moved. What is that thing that's moving?
The color.
Not just the color. The colors will be moving, but notice that there's something moving faster than the color. What's going to be moving faster than the color, than the dyes? What is the paper sitting in? That's number 4.
The salt water?
In our instance, salt water. But we also call it the solvent. So what would number 3 be then? If the solvent is moving up and it's sort of pulling the dye spots with it, what do we call the line which the solvent is moving? We have a special name for that. Nobody's read the instructions. Well, maybe somebody mentioned it on the chat. Let me see. Yes, John, solvent front, that's right. So number 3 is the solvent front. So that will be the distance from where we spotted our sample to how far the solvent has moved up our stationary phase. So that is the first distance we calculate, distance of the solvent. And that's the distance from the front, from where we spotted our sample. What's number 4 I've sort of given away. What do we call number 4? It's another word for solvent that's related to stationary phase. It's the other phase. What do we call the other phase of chromatography? One is stationary and the other one isn't.
The RF?
Nope, the RF, we'll get into the RF in a second. But number 4 is the thing that actually is moving up. It's the solvent that's moving up that's causing the dye spots to move. So if the paper is called the stationary phase because it doesn't move, what do you think we call the phase that moves? There's 2 phases in chromatography, one that doesn't move and one that does. Stationary and opposite of stationary. Take a guess what that might be.
Sedentary.
Sedentary is the same as stationary. It's the opposite of
Traveling.
that.
Traveling.
Traveling or mobile. Mobile phase. So we have two phases. One that moves, that's our solvent that moves through the paper or through the silica gel or through the column or whatever it is we're running. And the stationary phase is the one that doesn't move. Number 5 is where we spot the sample. And we call that the origin. Now, why do we make sure that we spot the sample above the solvent? Notice that the line across is not in the solvent. Why do we make sure that we spot the sample above the solvent and not below? In other words, we could spot our sample here. I mean, we do want to spot our sample on this location. We don't want to spot it in here. How come? That's a bad idea. That's a better idea. Why don't we want to spot our sample in underneath the solvent? What would happen then?
It's going to get mixed with the rest of the solvent.
Yeah, exactly. It's just going to dissolve in the solvent, and then it's not going to move up the paper. So the only time we want our spots, our dyes, to come in contact with the solvent is when they're already bound to the paper. And then they'll get carried up the paper by the solvent. If they're underneath, they're just going to dissolve in the solvent, and then that's it. Our experiment's basically over. So we don't want that. So everyone see the next slide? This is, really strange for me. So why don't we just measure the distances traveled? So if I'm doing the same experiment, we're all going to be doing the same experiment today. How come I can't compare my results to anybody else's? I can't compare them to Jean's, I can't compare them to Amanda's, I can't compare them to anybody's. I can't just measure the distance my dyes traveled and your dyes traveled and compare them. Why not? Like if my blue spot moves 5 centimeters, how come I can't compare that to anybody else's experiment?
Because their blue spot might have traveled a different distance.
Why? If we did the experiment the same way, how come their spot might move differently than mine? What's like the main reason why their spot would have moved a different distance than my spot? There's basically one major reason why that would be the case. We're all going to be using the same solvent. We're all going to be using the same paper, hopefully. So they should be the same, except for one thing. It's not the temperature. Temperature may affect it slightly, but there's something else that would affect it much more directly.
The size of the spot?
The size, that's a good point. If my spot is smaller, it probably won't have moved quite as much as someone else's whose spot is bigger. But the spot that's bigger is going to spread out more. That's when we talk about how, the actual physical doing of the experiment. That's something I definitely want to point out. When you make your spots, you want to make them as small as you possibly can, because they are not going to look like this. Believe me. You're not going to get these perfect, round spots. They're going to spread all over the place. But that's not the main reason. Something else would be the main reason why people's spots would move different distances. What's the main, think of what's the main driver in determining how far your spots are going to move? What's the main thing that's determining that?
Is it how pigmented it is?
Excuse me?
Is it how pigmented it is?
Yes, different dyes will move different distances. That is true. That's the whole point of doing the chromatography, the whole point of the separation. But if I'm separating exactly the same thing you are, why would your spot move different distance than mine if we're doing exactly the same thing? It's because I did something different than you. What would be the main reason that would make the spots move more or less? I'll give you a hint. It has something to do with this. It has something to do with that. Can you see the line I made there? Is that showing up?
Does it depend on how much solvent you have?
Not how much solvent, you're close. It's not how much solvent you use. It's something else involving the solvent.
The concentration?
Nope, because we're going to be using the same one. If we use different solvents, then yes. If I used, let's say, if I put 5% ethanol in mine, that would then be a different solvent, and the spots would move differently than yours. But if we're using exactly the same solvent, it has something to do with this distance here. What if I let my solvent front go higher than yours? What if I let it run for a longer time and the solvent front moved higher than yours did? My solvent front, I let it go 6 centimeters. You only let yours go 5. Would my spots have moved higher than yours or lower or the same? So if my solvent front is higher, so right here, I didn't stop you stopped your experiment after 15 minutes and I let mine go for 25 minutes. Say I forgot about it or something. I missed the timer. I let it go for longer. And so my solvent front, instead of being at this line, it actually went all the way up to here, a higher spot. Now, would my spots be in the same place as yours? Would they be higher than yours or would they be lower than yours?
Higher?
They'd be higher, exactly. So that is why we don't just measure the distances, because my spots could be in a different place than yours, but the ratio of how high my spots moved as divided by the total distance that the solvent went will be the same. And that's what RF means. What does RF stand for? What does RF mean? What's you're going to be measuring that, but what does it actually stand for?
Retention factor.
Right, exactly, retention factor. And how do we calculate that?
The distance the solute travels divided by the distance the solvent font travels.
Bingo, perfect. Yeah, it's basically distance of the spot divided by the distance of the solvent. And that should be the same for you, me, everybody. Even if everyone lets their solvent front move a different, maybe the temperature is a little warmer, things move a little faster. If things are colder, they move a little slower. You let yours run for 17 minutes, or let Ryan run for 18 minutes. Someone else lets them run for, half an hour. Whatever it is, if we all don't let it run off the top, and we'll get into why that's a bad idea. That's in your post-lab questions. If we stop it before the solvent gets to the top, that ratio should be the same for everybody. So does that make retention factor an intrinsic or extrinsic property? Do you remember way back when we talked about intrinsic and extrinsic properties? What's the difference between them?
Isn't one of them like affected by mass and the other isn't?
One of them is affected by amount and the other one isn't. So mass, is that an intrinsic or an extrinsic property?
Extrinsic?
Right, because I can have a gram of copper or I can have 10 grams of copper. They're both copper. So I can't tell, I can't say that they're different based on their mass, because the mass is just based on how much stuff I have. That doesn't tell me anything about the actual character of the substance I have. On the other hand, density does tell me something, because I got my 1 gram of copper, I got my 10 grams of copper. They're different masses, but they have the exact same density, right? Because density is an intrinsic property. Each substance has its own unique density, well, maybe not unique, but a different density. So is the retention factor an extrinsic property or an intrinsic property? If someone spots 50% more blue dye than I do, is their value of the retention factor for the blue dye going to be different?
So be intrinsic?
That's right. It's an intrinsic property. It's something we can use to identify the spots. So that's what we're going to be doing today, actually. So we're going to be using 3 different, I'm going to be actually using 4 different food dyes in determining what their retention factors are. Then we're going to look at different foods, and this you're given Kool-Aid as an example of something that has a food dye in it. And you're going to see whether or not that food dye that we looked at in the food coloring, those are going to be our standards, basically. We're going to calculate the retention factors for our food coloring, and then we're going to see if those same dyes are present in the food. Now, one of the things we're going to determine is, are their retention factors the same? And if the retention factors are the same, they're probably the same dye. What's the other thing we're going to be examining to see if they're the same? So we're going to be looking at these spots, and we're going to be calculating their retention factors, and then comparing it to the retention factors of the dyes in the Kool-Aid. So one of the things we'll be examining to see if they're similar is that, do they have a similar retention factor? What's the other thing that will tell us whether or not it's the same dye. It's another intrinsic property.
The distance it traveled?
Nope, because remember, the distance it travels is totally dependent on how long you let it run. So the distance it travels is related to our retention factor. So that, again, that's just part of the retention factor. So there's two basic properties you're going to be looking at. One is the retention factor, which is related to the distance. What's the second one? For instance, just look at this setup. I've got two samples here, well, three samples actually. I have sample 1, sample 2, and sample 3. So I am looking, I've got two spots here that appear to have the same retention factor, right? They've moved the same distance. The solvents move the same distance, so the retention factors are the same. What else about them tells me that they're the same dye?
The color?
Exactly, exactly. I might have two dyes that appear to be the same color, but the retention factors are different. Therefore, they are not the same dye. Even though they may look identical, if one of them is moved twice as fast up the paper than the other one, they can't be the same dye. Because if they were the same, they'd have the same color and the same retention factor. On the other hand, if I have two, let's look at this green one. This green spot is awfully close to the yellow one. What if I had two samples and I had a yellow spot with a retention factor of, let's say, 0.7, and I had a green spot with a retention factor of 0.7? Would they be the same dye? They have the same retention factor. Are they the same dye?
No.
No, they can't be because they're different colors. So those are the two things we're looking for. Are they the same color and do they have the same retention factor? They need to be both in order for us to say that it's the same dye. Okay. So instead of these like really pretty round perfect spots, you're going to get something that's going to look like this, right? It's going to look a lot uglier. In fact, I did this experiment last night with my 410 class, and you can see that you're not going to get these beautiful these beautiful spots. I'm hoping that my results are a little bit better today because I didn't know that I actually had the chromatography paper, the filter paper. So I used just printer paper. That's just printer paper that I like from my office. So, you know, it's probably not going to work as well. But you're not going to get these perfect spots. So for spots like this, trying to determine how far they've moved is kind of tricky. So what you want to do is basically see if you can determine where the middle of the spot is. The part of it that's the densest, that has the darkest color. So for instance, this yellow one here, the middle of the spot's probably going to be somewhere around there. And this blue one over here, somewhere like that. You're going to find, because these particular, you're going to be able to measure the solvent front with a fair amount of accuracy, because that's just going to be a line. But the spots are they spread out as they move. Not only do they move up, they sort of spread out to the sides as well. And so sometimes finding the exact distance the spot has moved is going to be kind of tricky. So we're going to want to find the middle of the spots and then take that as our distance, okay? So these are what we're going to be using for our paper today. And I found them, I didn't notice them the first time because they were sort of stuck between the Kool-Aid packages, so I missed it. But you should have filter paper that looks like this. Does everyone have these in that same bag? I think there should be, I think there's around 6 or 8 pieces of these. Now this is different from sort of regular paper. It's the same kind of paper we used to make the filters before. It's actually filter paper. And filter paper is something that has these, like, it's thicker and it has these large fibers in them, and it's really good at absorbing liquid. That's basically why they're good for filters. They absorb liquid and allow keep the solids behind. So does everyone have these?
Yeah.
Okay. So what you're going to want to do, just like it says over in figure 1, whoops, let me go back, is you want to cut these in half. So basically just, make a line at the top, make a line at the bottom, and then draw a line with a pencil. Make sure you use a pencil and cut them in half. Why do you want to make sure you use a pencil and not a pen? What would happen if you drew lines on your paper in pen?
It'll move when you do the thing later.
Yeah, your pen is, the ink in your pen is basically is a solution of dyes. I mean, that's what it is. In fact, one of the things you can do is, if you have a black pen, you can use one of these, put a mark with a black pen, and black pen is beautiful. It's got like about 6 different colors in it, because basically black is all the colors mixed together, right? And so black pen, when you separate the colors, you'll see it's really pretty. You'll get like red and blue and green and yellow. They're all there. So you definitely don't want to use pen. Pencil has like a tip made of graphite, right? And graphite is just carbon all linked together. Those are very large molecules. And so they're not going anywhere. They're not going to move. They're too big. The molecules of graphite are too big to move. So they just stay put. So use pencil. They're not, those lines aren't going to move. So at the bottom, you want to have, here we go. You want to have a mark 1.5 centimeters from the bottom, and then draw a line across. That's going to be the origin line, okay? Also from the bottom, make another line 9.4 centimeters from the bottom. So there's the bottom, 9.4 centimeters, draw another line. That's where we're going to put a pin so that our strip doesn't touch the bottom of the tube. We don't want the strip coming in contact with the bottom. So it will just basically sit just above the bottom. All right. Those are the 2 marks we want to make. And then on the bottom where we've made our mark at 1.5 centimeters, just make a little point midway in between, and then just make a cut from the 1.5 mark to the middle, 1.5 mark to the middle, so that you have this point. This will basically, this point is where the water will start moving up. Okay. So take some time to make. I'm not sure if you've made those already. It doesn't take very long. It takes like about 5 minutes to do. So now you can either do all of this experiment at once, or you can do some today and some tomorrow, because you're going to have to make a lot of these things, because we're going to be doing 3 different food colors, and then the 3 different Kool-Aid colors as well. So we're going to be doing 6 different ones. So you can do the standards with the food dyes today and do the Kool-Aid tomorrow, or you can do it all today at the same time. I'm going to do the 4 food dyes that are supplied, and I'm going to do one of the Kool-Aid to see what it looks like, and then we'll go over how to to work with the data. So I'll give some time between like the end of this and starting the experiment so you guys can get all your stuff together. So for the Kool-Aid itself, weigh out 2 grams of the powder, add 5 mils of water, and just get it all dissolved. You're gonna want these things to be super concentrated, because as you can see, these actually started with like a tiny spot. And it's kind of hard to believe, but like a really, really small spot. And look how much it's spread out. So these things are going to spread out. And as they spread out, they get less and less concentrated. And so not as easy to see. So you want these things to be dark, like really, really, really dark, because as they move, they're going to spread out and get more and more diffuse, and the color will get more and more diffuse as well. And when we're starting the experiment, I'll show you how to do the spotting. It's really important to do the spotting properly. All right. So what we're going to be using the spotting with is that in the bag with all this other stuff, we got these transfer pipettes. But these transfer pipettes are different from the ones we've been using. Let me grab one. So these are the transfer pipettes we've been using in the past. So you can see the tip on them is kind of wide. So we got the bulb here, and then there's the end that we've been using to transfer liquid with. This is the transfer pipette that comes in the comes with the Kool-Aid packages in the bag. Notice that the tip of this is much smaller, much smaller. So what we're going to do is cut it about, I don't know, there, give it about like 4 or 5 centimeters, and just cut it off. And then what we're going to do is we're going to use this to spot our samples with, because we want the samples to be super, super small, as small as we possibly can. And so what we're going to do is pick up some of the dye, not a whole lot, spot it, let it dry, come back, spot it again, let it dry, come back, spot it again, let it dry. And each time we spot it, we don't really want the spot to spread out anymore. So we basically try to keep spotting it right in the middle each time, and keep that spot as small and concentrated as we possibly can. Okay.
When you mark the little papers, does it have to be exact?
Do we do what?
Does it have to be like super exactly like 1.5 centimeters?
No, I mean, if it's 1.4 or 1.6, it's not going to be any big deal. No, because as long as it's a straight line, because that's what we're going to be using to measure our solvent distance and our spot distance. That's going to be the origin. So we measure from there. So make sure it's a straight line across. It's a little tricky getting all those strips together and then trying to draw a straight line across all of them without them moving. Believe me, it's kind of tricky to do. But yeah, just take your time and draw a nice thin straight line with a relatively sharp pencil. All right. Then our solvent is going to be water with 0.1% sodium chloride, so not a whole lot of sodium chloride. So we're going to have 100 mils of sodium chloride and 0.1 grams, or sorry, 100 mils of water and 0.1 grams of sodium chloride. So a really, really, really small amount. And then we're not going to need much of them either, because we're only going to be putting about 1 mil in each tube. And you can use these transfer pipettes to measure that, because, I don't know if you can see it very well, we're right below, if you look at them, right below the bulb, it says, can you see that? It says 1 mil. Okay, so if you drop water just above that 1 mil mark and then just squirt it all out, that's a mil. And so that doesn't need to be super precise either. If it's a little less than a mil or a little more than a mil, that's fine. You don't want it to be like 3 mils, because then the water level will come up past your spot and then your spot will just dissolve in the water. So you don't want that. All right. So, go. So this is, so the data table 1, can you see the data table 1 here?
Yeah.
All right, so it says known F, D, and C dyes. What's F, D, and C stand for, do you think? Take a guess. Basically, these dyes have been tested to see whether they're okay for, human consumption. So what do you think the F stands for?
Food.
Food, right. What does the D stand for?
Drug.
Drug. And the C? C is a little tricky. C I had to look up.
I don't know.
So when will you come into contact with dyes? You'll eat them. You'll take them in the form of a drug. What's the other way you'll come into contact with something with a dye in it? You need to worry about.
Chemistry?
What is it?
Chemistry?
Chemistry? No, something you would use, like, I don't know, just on a semi-regular basis, where your body comes in contact with something that might have a dye in it. Cosmetic, that's right, John. Man, John, you're crushing it today. Yeah, cosmetics. So it's been tested for foods, dyes, and cosmetics. That's what the F, D, and C stands for. So these are so table 1 will be the food dyes that come with the package, and there's 4 of them. I don't know why we're just not doing all 4, but we're just using 3. We're just using the red, yellow, and the blue. I'm going to use the green too, because it seems that the green separates into the yellow and the blue, which you would expect it to, right? So you take. We're going to be spotting these dyes. Obviously, you're going to be looking at the color of the band, which should be same color as this. You're going to be measuring the distance that it moves from the origin. So if this is my strip, there's my origin. There is the solvent front. And there's my spot. So this distance will be my D spot. That's how far the band traveled. This distance from the solvent to the origin, that's my D solvent. And then the retention factor is just going to be D spot over D solvent. And you'll want to measure these in millimeters, because you'll get the most accurate measurement there. And as far as retention factor, I mean, you'll have three digits for the distances, but then it's the measurement of the distance for your spot will be so inaccurate that, as far as RF, probably 2 digits is more than sufficient. So don't bother giving a retention factor of like 0.693 or something, because there's no way you can measure it that accurately. So if the units for the distance is going to be millimeters here and millimeters here, what are the units of RF? What are the units for retention factor?
Is it in a percentage?
Nope. Well, in a way, yes. Yeah, in a way it is a percentage. But what unit would we include with it? Because whenever we do anything scientific, we always have to have a number and a unit. What would be the unit for retention factor? Remember, whenever I do a calculation, I always want you to include the units, right? So if we include the units for this calculation, what do we wind up with? John comes through again. Yeah, there's no units. There's no units because there's millimeters on the top, millimeters on the bottom. Those cancel out and there's no units. It is just a ratio. So yeah, you were right as far as it being a percentage. It's just a ratio. So there's no units. Very good. Okay. So that's the first data table. And then the second one is for the unknown mixtures, the Kool-Aid. And if you have, some time, you can try pen or they suggest, I think, M&Ms, because M&Ms have like a really brightly colored shell that definitely has some food dye in there. And so you will be able to identify the dye, as we mentioned before, by using two criteria. One is the RF, and the other one was what?
The color.
The color, right. So our first criteria is the retention factor, and the second one is the color. So we want to make sure that both of those are the same in order for us to say that that is definitely this particular dye. Okay. Now, so what I'll do is give everyone like 10 minutes to get all their stuff together. And then we'll come back and we'll start. Are there any questions before we get started? Things I may have left out? Basically what we're going to be doing, what our separation is, why does one spot move faster than another one? What we're looking at is something called differential affinity. What do I mean by affinity? When a molecule has an affinity for something, what does that mean?
Like an attraction?
Exactly. It means basically if the red dye moves faster up the paper than the blue dye does, it means the blue dye has a higher affinity for the paper than the red dye does. It means the red dye has more affinity for the solvent. So as the solvent moves, it carries the red dye with it because the red dye is bound more closely to the solvent. If something doesn't move very much at all, it means it's more tightly bound to the paper than it is to the solvent. And that's how all chromatography works. We're basically looking for which substances have a greater affinity for the stationary phase. They're not going to move much. And which substances have a greater affinity for the mobile phase, and they'll move more. And it's all related to the intermolecular interactions you're sort of learning about now. Hydrogen bonds, van der Waals interactions, all those different forms, dipole-dipole interactions, ion-dipole interactions. That's one of the reasons why we're including the salt, is so that the solvent will have ion dipole interactions, and the paper which is basically sugar, right? Paper is just made of polysaccharides, made of sugar. So you'll have dipole-dipole interactions, but you won't have any ion dipole interactions. So that's why we include the salt. So all those different interactions are taking place, and we want to see which ones are stronger for each of these individual dyes, and that's why they separate. So any other questions before we get everything together and start? Is everyone clear on what we're going to be doing today? When we actually get everything set up, I'll go into detail, like how to spot these things. Okay. So 10, then we'll get started. I'll give you time to get all your stuff together. All right, cool. So I'll see you in 10 minutes. So you should start by making your solvent. So the solvent is just 100 mils of water in a 250 mil beaker, and just add 0.1 grams of sodium chloride. So just, just some salt. You probably got some salt at home. It's not a lot. So don't worry too much about getting exactly 0.1. I mean, if it's, 0.12, it's not a big deal, but around 0.1.
Does the water have to be a certain temperature?
Room temperature. Yeah, you don't want to, I don't think you want to use super hot water because then that will move like way too fast. So just room temperature water.
It's like a little warm. Should I put it in the freezer?
I mean, how warm is it?
Like, not hot, just like not room temperature though.
So by the time you've got everything spotted, it probably will have cooled down. So it probably won't be a big deal. And then put a mill. So remember that line just below the bulb is 1 mil. So a mil should be about that much. And you can write on these on the top of the paper. So, R for red or whatever it is you're doing. And then these are the pins we're going to be using. They came with when we did the experiment on looking at different metals to see if a reaction series, to see if one metal would replace another one. We got these pins in there. And so I think there's about 7 or 8 of them. So we're going to be using those. So have those nearby. All right. So it's also going to mix up my Kool-Aid as well while I'm here. So I want 2 grams. Okay. Try not to make a huge mess. All right, 2 grams. That's going to be quite a lot. Okay, there we go. 2 grams. So 2 grams is about that much. Not a whole lot, but it should make for a very dark mixture. It's going to be a lot more concentrated than the typical concentration we would use when we're drinking these things. So I've got some filtered water, 5 mils of filtered water. Five. A little bit more. Here we go, 5 mils. Yeah, basically this is just going to be black. We won't know what color this is. You can definitely smell that sugar though. And it'll probably take a minute to get this dissolved because that is yeah, this is black basically. I think we just stir it until you no longer hear the crunching of the sugar in there. Is just the 3 of us working on this today? Ruby, you're not going to play?
No, I'm just watching.
Oh, come on, man. Oh, man, still a bunch of sugar in here.
would construction paper work as paper? Because it feels like the filter paper, and it's like it looks like
If it's white, yeah. If you have just like white construction paper, that'll probably work fairly well.
okay.
So the width of it, it should be about 1 millimeter in width. That fits in the tube. So if you could cut them to about 1 millimeter in width,
Yeah, I'll try to make them the same size.
yeah, and the length won't be. You just cut the length to whatever you want later. All right. I'm not hearing crunch, crunch, crunch of sucrose anymore, so I think it's probably all in solution. Man, does that smell strong. Ooh, already got my first mess. Oh, let me get something to wipe that up. I should have already had some of that. Okay. And I may have a permanently blue thumb too. Okay. So suggest that you, with the food dyes, you just get like a piece of piece of tin foil or something like that, and just squeeze it. Just squeeze out a couple of drops, because that's really like a drop or two is all you're going to need. So I need to get my dyes. So here's my dyes. So I'll go. So I got my red, my yellow, my blue. Where's my green? Oh, here's my green. So you just squeeze them out onto the right. So there's my red spot, and then my yellow. You see the yellow is like so concentrated that it actually looks red, but sort of this dark pink color. The blue is going to look black, and the green is going to look black too. So then you're going to want to use the same tip for spotting. You're not going to want to mix them up, because if you mix them up, then you're going to have more than one color in there. So I want to have these arranged. So red, yellow, blue, green, and then my Kool-Aid. So I get my strips together. And you're only going to be taking a very small amount of sample each time. So don't worry if it seems like you're not getting a whole lot. Not getting anything drawing out. There we go. So just get a little bit drawn up in the tip, and then try and put it in the middle of where you have that line, the one just above the pointed end. Oh, that's spread out quite a lot. So you can see how much that's spread out, right? You're probably going to want to keep it smaller than that. So I'm just going to let that dry. And then I just put my tip down to the color I used. Then get another one. Then go for the yellow.
Use a different strip every time, right?
Definitely. And you can have one where you mix them all together so you can see them all separated. You can definitely do that if you'd like. But yeah, the whole point is to try and keep these spots as small as possible. So just give it a little touch and then take it away. A little touch, take it away. So there's the yellow one. So the yellow one is a little better than the red one. It didn't spread out quite as much, but you want to get these as small as possible. Because as you saw from my results from the one last night, these things spread out tremendously. There's really not a whole lot we can do about that.
Maybe on the line or above.
You want it right on the line. Yeah, but you want to have like the middle of the spot to be right on the line. Oop.
And then in the thing, it says that they want the spot 3 millimeters in diameter.
That's what you're shooting for.
So like keep adding more dye.
No, if you're smaller than that, better.
Oh, okay.
Just basically let it dry, and then we're going to spot them again. We're going to spot them at least 3 times. Having trouble getting the blue to come out. There it goes. Oops, a little too much. My green. Yeah, it's really hard to get these to be super, super small. I mean, you want them to be, but sometimes it all comes out at once.
And about how many times do you like spot it?
I'm going to spot each one 3 times. So I have a nice dark spot. No, now I'm having trouble with the green one. If you're having trouble coming at it, you just give them a little flick, try and get the dye down to the bottom. Come on. There we go. And then my Kool-Aid. I can actually see it's purple now that it's spread out a little bit.
So So do the spotting of the food coloring and the spotting of the Kool-Aid at the same time.
Yeah, I start one and then I move on to the next one. So by the time I'm done, I can then move back. The first one should now be dry and I could respot the red. And just go right back to the center. Oop, there we go. That red one is going to be really big. And spot the yellow one again. Yeah, these spots are definitely bigger than 3 millimeters, I'm pretty sure. That's the ideal, but it's going to be tough to get them that small. Okay, there we go. Whoa, dear. Oh, my green one. You can see that I really overspotted the green one. So that one's going to be tricky. Luckily, that one wasn't even recommended to be used. So that's adding too much. That'll make a nice mess, the green. Okay. And then just one more time through. Okay. Yes, this blue one looks pretty good. So I think that's a, I don't know if I'm going to be able to do much better than that as far as spot size. So then the other thing is you're going to want these to dry before you put them in the test tube, because you want the dye to fully be absorbed by the paper before it comes in contact with the solvent. Because if it isn't, then again, the solvent is just going to, it's just going to dissolve in the solvent and then move up with the solvent. You don't want it to do that. The only time you want the dyes in these samples to come in contact with solvent is when it is in the paper itself, not outside of it. All right. So just give these a couple minutes to dry. So then put 1 ml of your salt water in each one of the test tubes, however many test tubes you've got. The pin is going to go where this line is. That's the line you made at about 9.4 centimeters from the bottom. Push the pin right through the middle of that line without, stabbing yourself. Here we go. So right through the middle of the, and then when this is dry, we just take it over to the test tube and drop it in. And what should happen is that this spot should be above the solvent. Let's hope it is. It looks like I could, well, and you don't want the paper to be touching the side, which is a little tricky. Because I think there's some static electricity that's, there it goes. You see, it's just making contact with the, it's already moving.
So it's supposed to touch the water or not supposed to touch the water?
Well, it has to touch the water, right? Or else the water's not going to move up the paper. So, but you want that tip, that sharp tip, to just be making contact with the solvent.
So like this?
Perfect. That's exactly what you want. Yep. All right, so here's my yellow one. Here we go. Yeah, see, it's just making contact. That's precisely what you want. And the other thing you don't want to do is once these have started, you don't want to disturb them. Like you don't want to shake them. And then it suggests, how long does this suggest for us to let them run? Anyone remember?
It says for about 15 minutes.
Right. But basically you want it to move up about 3/4 of the way until it reaches 3/4 of the way up to the pin there. And that's, the longer it runs, the better separation you're going to get. But you don't want it to let it run so long that the solvent runs up the top of the paper. Oh man, these things are moving fast. Yeah, these move much, much faster. The printer paper that I used last night, I could let them run for like an hour and it still hadn't moved halfway. I think the printer paper is much, much smaller fibers. It's much denser. But these things are moving really fast. So you have to keep your eye on it. That red one I just put in the water like, I don't know, 3 minutes ago, and it's already like halfway up. So if they run for different times, not a problem. You can stop one before you stop the others. Because remember, we're not looking at the distances, we're looking at the ratio of the distances. Yeah, I mean, you can see how fast this red one is moving. It's moving tremendously quick. So
we don't really have to keep track of the time, like we don't have to record it.
I mean, you probably should, but I wouldn't. What I wouldn't do is like set a timer for 15 minutes and then walk away, because it looks like some of these are moving a lot faster than I expected. So, oh man, the Kool-Aid's looking really nice. So you can already see with the Kool-Aid how it's separating. I used grape, so purple, so we would expect red and blue, and that's pretty much what we've gotten here. So as long as your spots are relatively small, this should really work fairly well. Although, like I said, these spots are going to get pretty big. Yeah, I mean, the blue was my smallest spot. You can see how much it's spread out already. So, I mean, these things are going to spread. There's not really a whole lot you can do about it. So then when they're done, you just take them out, and you're going to want to take a pencil and mark where the solvent front is, and then just let it dry. And then you can do all your measurements when it's dry. So I think I have my pencil over here. Yeah. That's why it's good having a kid in elementary school. There's always lots of pencils around. Don't really have much need for them anymore. So what's going to be tricky is when you have something like this that's spread out so much. It's basically what you're going to have to do is take like where the darkest part is. You see this is a super long tail on this thing. So find the darkest part of the spot, circle it with your pencil, and then go draw a line through the middle. That's basically what you're going to have to do.
And then that would be your solvent front.
So your solvent front is where the last place the water moved. So with this, man, this is looking great. The Kool-Aid one's looking awesome, looking better than the dyes actually. I probably spotted them too much. Probably one spot would have been sufficient. The solvent front, it's kind of tough to see from here. If you backlight it, if you look at it against the light, it's much easier to see. So hold this up against the light and you'll see that it's a little bit darker above these two colors. That's the solvent front. Do you see that? If you hold it up to a light, do you see that it's a little bit darker above it? That's the water moving.
So it's like where the water ends.
Yes, exactly. Yeah, we probably should have just spotted these once, because man, these dyes are dark enough that a super small spot is really what we're looking for, because they're so dense. So this one's almost done. I mean, probably let this go for like another minute or so, then I'm just going to stop it. But with this one, the solvent front is basically at the end of the color as well. Because I think there was just way too much dye in that one. Now see, the green is separating as well. I've got the yellow below, and it's still kind of green, but it's a little more blue on top. But yeah, the best looking one so far is definitely the Kool-Aid. That looks, I'm getting the sharpest separation. Probably because I didn't overspot that one. How other people's looking? How's yours looking, Lance? Looking all right?
Yeah, it's looking all right so far. My blue is like the one that's moving the fastest.
Yeah.
Is it supposed to look like that? It's like on the side of the paper, as opposed
Oh, yeah. If it gets to the side, it's basically going to run up the side. That's pretty much what happened to my red one here. It's so hard to get this, because basically you want, oh, that's really nice. That's really good. Yeah. Yeah, you want the smallest spot as possible. It's really tough to do, to get it super, super small.
So for like this one, you measure where the
Oh,
purple.
that's perfect. That's really good, Arissa. Yeah.
You-
Is that your Kool-Aid one?
Yeah, that's the grape one.
Yeah. Yeah, I'm getting the best results with that one too.
That one got in my mouth and it's very sour. And
Gee, how did that happen? It's sour.
'cause I was
How's that
stirring it,
possible? It's all sugar.
I was stirring it and it went everywhere. And now my
Uh,
desk is all purple. So
All right, so I'm going to stop this red one. I'm going to pull this one out. And just take the pin out and just let it, put it back on the paper towel. And then while it's still wet, just very gently with the pencil, just mark where the water was. You don't want to mark it, don't mark it, because right now it's wet, so it's very easy to break. So just put a very light line across it. Yes, just a very light line. I don't even see that, but just a very light line. Because if you do it too much, it'll just break.
you make the solvent line now, and then when it's dry, you make the little like
Yeah, exactly. Mm-hmm. Take my yellow one out. Mark where the solvent is.
Is it normal for like the solvent line to be the same as the color line?
For some of them, yeah. I think because yeah, I think I just way overdid it with the red. And so, yeah, basically it's just a streak all the way up, and the solvent front is basically where the color ends. And so you can always repeat these. You have more paper. So you could try it again with like a single spot. Because yeah, these are pretty intensely colored, so you should be able to see it all right. And if your solvent front isn't straight, just draw it the way it looks. Yeah, the blue definitely runs the furthest. So the blue should have the highest retention factor, because it's basically running with the solvent. You can just like yours, Lance. Looks the same. So it looks like blue runs the fastest, and then red, and then yellow. And then, yeah, that Kool-Aid looks awesome. Yeah, you can see that with the Kool-Aid one, we've clearly, I mean, that's a really nice separation.
This
We've
one's like,
clearly separated them. What have you
The-
got there, Arissa?
this is the orange one, but it's like pink at the bottom now.
That's your yellow?
Mm-hmm. The orange Kool-Aid.
Oh, the orange. I didn't try the orange one. Man, what's strange is that the blue and the red on the purple Kool-Aid look to be in different positions than the blue and red of my other dyes. The blue moved much higher, and the red moved much higher as well. Interesting. How did the strawberry Kool-Aid look? You did that one too, Arissa?
Yeah, it looks subpar. It's not that nice.
Oh, it just kind of just all spread out. It was
Yeah.
just looked like a single color though, right?
Yeah, kind of. It's like a
Is there a little bit of yellow in there?
no, it's like a gradient. Like it goes from pink to red.
Yeah, so it looks like a single color.
But
Which
yeah,
makes sense, right? Because they just want red for strawberry.
this one is the like most colors.
Yeah. All right, so I'm going to move back to the table. All right, so any questions about the lab, how you do the measurements, anything like that?
Oh, for last week's lab, I got a really high heat capacity for my calorimeter. Is that normal?
How high is high? Too high? What was the
It
difference
was
between the hot water and the cold water? Was it a big difference?
from like the zero?
So let me put up a. So when you calculate it, Q hot and then Q cold, was there a big difference between them?
Yeah, it's around, well, for one of them, it was way bigger than that. Well, no, one of them was like 3,000 joules, and then the other was like 1,200 joules.
Oh, that is a big difference. Yeah, I'd go back and look at that, because I think the difference should only be around 10%.
Okay.
Yeah, there shouldn't be that big of a difference. Like you should have something like, if you had like 5,000 joules and 4,100 or something like that. That would be something that'd be a difference you'd expect. If you have a difference of like more than 2 to 1, that's kind of odd. What was the
So
difference
the
in
Q of,
temperature? What was your delta T for each one? That'll help us figure it out.
24 degrees Celsius.
So,
Then
so the starting
19
temperature
degrees
was
Celsius.
24?
No, that's the delta T. That's the temperature change.
Oh, for which one?
For trial 1, it was 24, and then for
No, for
trial
hot
2,
or
it was
cold,
19 degrees Celsius.
they should have different delta T's. Which one was different? Which one was Q cold? What was the
Oh, that's
change of the cold?
cold.
Sorry?
That's for the cold.
Oh, so the temperature went up.
That's for the cold.
Okay. And what did it go down for the hot?
It's not in my data table. It's in my notebook. Let's see, temperature. It's about like
So
30.
10 minutes.
Yeah, around.
So it went down 30. Okay. So, yeah. So you should see everything else is the same. So they each had, you had 50 grams of each one, right? Because there's 50 mils. The heat capacity of each one is 4.184. And so the difference times minus 30. And then the other one is also 50 grams times 4.184 times 24, minus 30 and plus 24. So the difference should be 6 degrees times 50 grams times 4.184. That's the number of joules that went into the calorimeter. So that's 300 times 4, so that's about 1200. And then you divide that by the change in temperature that went up, 24. And so that's what, like 50 joules per degree.
So that's the
Which
difference.
is a little high, but it's not outrageous. I think I got like 25 or something.
Okay, so that's the difference between the two heat capacities.
No, between the two heats. Between the two heats. There was this much heat was lost by the hot water. This much heat was gained by the cold water. And the difference was the heat that was gained by the calorimeter, which would be this. And then we just, whatever the difference is. And then we just divide that by the temperature change of the calorimeter, which is the same as the temperature change of the cold water. It went up 24 degrees. So the calorimeter went up 24 degrees as well.
Okay.
Okay?
Mm-hmm. Thank you.
Yeah, so I mean that's not that's typical. That's what you would see. So the drop in the hot water is going to be a little higher than the rise of the cold water. If the calorimeter was perfect, then each temperature change would be exactly the same. The hot water would drop 30 degrees, and the cold water would rise 30 degrees, but it isn't exactly the same. It should be a little different. So about 6 degrees or so, that's to be expected. Okay?
Okay. Thank you.
No problem.
Mm-hmm.
So anybody else have any questions about the lab today or anything else? I see something on the chat. I do accept late lab reports. I do indeed. And how many points I deduct depends on how terrible they are. All right. Any other questions before we call it an afternoon? All right, everybody. Have fun with this one. Yeah, keep your spots small and keep an eye on where how high the water gets and your solvent level. Okay. All right, everybody. I'll talk to you soon.
Thank you.
All right.
Thank you.